it's a great pleasure to introduce Bernard Crespi, who's our speaker today. He's a professor of evolutionary biology at Simon Fraser University. His research focuses on a huge range of questions in evolutionary biology. A lot of you know Bernie, and you know that this is true. This range of questions includes the evolution of social behavior, human health and disease, including mental health, evolutionary conflict, trophic interactions, and also adaptive radiations. He's really known for asking big questions, and he's also known for shining new light on these questions, really, and often an innovative and synthetic way, bringing together lots of different lines of evidence. So with that, I'll turn things over to you, Bernie, and you can share your screen.
Thank you, Charles. And I wanted to thank you and Meredith for giving me the opportunity to be here today and for running this program. And thank you everyone for turning up today. Everybody got that screen? Is that visible properly?
So we see your presenter mode with your notes and next slides.
I'm sorry, what's that?
We see your presenter mode,
okay.
so we see your notes and next slides.
I'm gonna go to let's see. So I want this to be bigger, right? Full screen.
What happens if you click on Use Slideshow? Does that switch it? No?
It just kicked me out. There.
Yep.
Yeah, that's perfect.
Okay. All right. One of my favorite books is The Devil's Dictionary by Ambrose Bierce. So I thought Darwin deserved his own dictionary, so I made one up. And the first entry that I put in there was something called Batural selection, which is, by my definition, natural selection associated with infectious diseases in or from bats. And that's because I see that as kind of a special form or arena of natural selection. Today I'll be talking a little bit about bat life histories and immune systems, bat-derived diseases, putting this all in an evolutionary medical framework, talking a little bit about virus strategies and tactics, and putting this all into the context of the current epidemic, epidemiology, symptoms, treatments, evolution, and then a few health-related implications and research implications towards the end. So this is the overall framework. The idea here is to take these various diverse lines of independent evidence, from bat life history to bat immune systems to human immune systems to COVID epidemiology, symptoms and treatments, and coronaviruses, and take all these pieces of the puzzle and try to fit them together into some sort of convergent, concordant, or coherent picture in an evolutionary medical framework. And I'm not trying to answer any questions today. I'm trying more to develop some specific hypotheses that are testable, that can help to direct the collection of data. So first, a little bit about bats. If you wanted to design a mammal that was good for viruses, that wasn't humans, you'd probably come up with something a bit like bats. They live in large, dense populations, a lot of opportunity for transmission. They have air travel like us. They fly around at night, and they also migrate long distances. And they kind of live indoors like us in places with amenable temperatures, humidity, and low ultraviolet. Taken together, this means that bats are expected to have been subject to a long history of strong selection by viruses. And we see evidence for this in some of the molecular changes at the origin of the Chiroptera. Second, bats are unusual because they fly. When they fly, their temperatures go way up, very high metabolic rate, generate a lot of reactive oxygen species. They having to deal with this is apparently selected against the use of inflammation in bats, because inflammation basically cannot be tolerated if you need to be flying around. Consequences, bat viruses evolve to withstand high temperatures, so a fever defense probably doesn't work as well. They go up to 105, 106 degrees when they're flying. And bats have been selected to express low levels of inflammation as an immune defense. So they're very different from us in this regard. They use some other means of defense against, for example, viruses. And they live a long time, so viral infections can be persistent. The long lifespan is probably related to their low levels of inflammation as well. Very brief overview of bat immune systems and how they're different from ours. Bats rely considerably on high interferon activity. It's constitutive in some of them. It's very highly activated, induced in others. And this activity serves to limit viral replication. And bats, this seems to be one of the main strategies that bats use to keep the high levels of virus to which they're vulnerable under control. They also, as I mentioned, express low levels of inflammatory responses. You can see this in some of the genes that they've lost and that they have. So they have less collateral damage from inflammation in the innate immune system, and that's good. But if you don't have this defense, this may make it more likely, and this has been suggested as well, lack of inflammation making it more likely that viruses can persist in the long term at relatively low levels in bats. There's various other differences between bats and other mammals with regard to their immune systems that I won't go into that are important here as well. We're going to focus mainly on what seem to be the biggest ones, high interferon and low inflammation. Here's an example from SARS-CoV-1, list of their different proteins and functions, and you can see 8 of them here that are specifically adaptive in the context of antagonizing interferons. Probably most viruses do this, go after the interferons. This is a pretty normal thing for viruses to do. The coronaviruses seem to do it more. One way that they may be able to do it more is that they have a large genome for an RNA virus. This is probably because they've evolved a form of DNA repair, proofreading function, which other RNA viruses don't have, allowing them to have more genes. And if they have more genes, they have more weapons, essentially, that can antagonize the interferon system of their hosts. And there's some evidence from the SARS-CoV-2, they induce a weak interferon response in us. And for example, their ORF3b gene is a good interferon agonist. And for SARS-CoV-1, people have a reasonable idea of how these anti-interferon genes are working. Each of these little red bits of text and blocking arrows is somewhere where one of these viral proteins is interfering with the interferon system, which is important very early in the response of the host to the virus. The interferons are involved in getting the immune system to kind of wake up, activating cytokine responses. This gets transduced over to the brain to activate sickness behavior and make you feel sick. And then this also links over to the adaptive immune system and the induction of this whole system and the generation of eventually of memory cells, and that might be important there. So if you're interfering, if you're a virus interfering with interferon, you're going to be, if you're specializing in that, then you're going to have differential effects on the initial turning on of the immune system. Okay, a little more about bats. For the reasons that we talked about earlier, apparently many zoonotic viruses are from bats. Bats appear to have a lot of viruses. There's some controversy about that, but they certainly have many viruses that are zoonotic and get into people. That's the purple down here, and there's the coronaviridae there. And the bat-derived viruses in humans seem to be especially bad. There's good evidence that they have high virulence, high case fatality rates, and we'll be talking about some of the possible reasons for that in just a couple of minutes. So here's phylogenetic distance from humans and case fatality rate getting very high in some of these bat-derived viruses. So why is that? Is an important question. Some basic evolutionary medical principles: mismatches, conflicts, and trade-offs. Mismatches are obvious. We have a bat virus. It's not adapted to the human immune system, and this may give rise through various mechanisms to high virulence, potentially in some cases. And of course, we have conflicts and arms races, well documented in many virus-host interactions. Two main strategies of viruses: hit and run, acute infection, jump to somebody else before the immune system kills you, or it's like influenza, whatever. Hit and stay is a situation where you get a virus and the virus becomes chronic, stays present at low levels. We can think of this as tolerance. And this seems to be what happens in bats. You can detect many viruses which are particularly nasty in humans. You can detect them in bats, and the bats show no evidence of being sick. Whole variety of tactics in the conflicts here. Trade-offs: inflammation is good because it's fast, it's bad because it's non-specific. It's good because it kills virus, it's bad because it also damages you. As long as B is greater than C, you can select for the inflammatory responses. Trade-offs with adaptive immunity: it's good because it's highly specific, little smart bombs. It's bad because it takes a while to kick in. It's especially useful in early midlife. By the time you're getting old, the odds that you're going to get hit by a virus, the same virus twice, are getting very low. So when you're older, the adaptive immune system is becoming less useful and less effective. So here's the main hypothesis I'm going to be talking about today. It's based on mismatches between humans and bats with regard to interferon system and inflammation. So you get a coronavirus in a bat. The bats have high interferon and low inflammation. The virus is controlled, but it's not eliminated. It can persist, and it's tolerated. And there's good evidence for this toleration for many viruses in bats. And then the question becomes, what happens when you get the virus in us? We have a relatively low interferon level of defense compared to bats. This may allow the virus to build up to higher levels than in bats. And because the interferon system is involved in kind of turning on the downstream systems and turning on sickness behavior via the brain, we can get a delay in activation of the acute phase responses, and this can lead to transmission by people who are pre-symptomatic. which, as we've seen, is one of the main issues having to do with high transmission of this particular virus. So the virus, by this hypothesis, can build up to relatively high levels because it's got an especially good anti-interferon system. Eventually, the immune system kind of wakes up and says, uh-oh, and you got very high levels of virus by then. Immune system turns on high levels. Coagulation defense system turns on very high levels, detecting a high viral load, getting severe pathology in vulnerable individuals. So what we have then is the mismatch between host defense and viral offense that is two-pronged. It's a function of the interferon system and the hyperinflammation system working together. It's a nice picture of what might be going on by Jamalu in the preprint server. Mild disease, virus building up here, pre-symptomatic people, and eventually you get the interferons turning on, reducing the virus. You get an inflammatory response that helps out to build up all your white cells, and in most people you end up getting rid of the virus. However, in other people, we'll talk about which people and why in just a minute, the interferon system is relatively delayed. You can build up to a really high viral load. You get the inflammatory system turning on at a high level, coagulation system turning on at a high level, and all the sorts of pathology that you're seeing in the lungs and other places do appear to be related to hyperactivation of inflammation and coagulation defenses. Now, what does this mean potentially as regards treatment? Well, it means that early on in the infection, you want to treat people with antivirals as early as possible, okay, to try to prevent this early buildup. You want to get the system turned on, perhaps treat with interferons themselves. Can you do that early enough to make that big a difference? I don't know. That's an empirical question. Antivirals early, and then later on in the severe cases, anti-inflammatory drugs. And you don't want to give the anti-inflammatories early, and you're not going to gain by giving the antivirals later. It has to be a sort of a staged approach here. And this idea for the inflammatory part fits with the recent success with dexamethasone in treating severe cases. So the next question is, why do some people have mild cases and other people have severe cases? Well, here we have case fatality rates by age. So very low, going up, 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 up. And then the question is, well, okay, what is this all really associated with? What is the best correlate here? And it turns out that if you take the normal curve of all-cause mortality in humans and lay it up against the case fatality rate curve of the virus, then you get a pretty good match here. It's just going up. These faded lines are all-cause mortality in humans, and they seem to track the case fatality rates quite well. And this was also found to be true in Black Death from digging people up in the cemeteries and determining their age. So there's a nice match here between all-cause mortality and COVID-19 death rate curves, suggesting that age itself, something about age itself, is fundamentally important. But what about the gender bias? Very good evidence, strong male bias in mortality from COVID-19. Okay, how does that fit in here? Well, it turns out that if you look at the sex-specific mortality rates by age, the male mortality rate seems to match the mortality rate of females who are about 10 years older. Okay, so females here matches males here, females here matches males here, and so on, and so forth. So what this means is, if we look in the context of male and female mortality, is that, well, we do have evidence that males age, pace of aging is faster in males than females. This ties in with males dying on average 8 years earlier than females do. So the bottom line is that, yes, there is these sex differences in mortality, but they might largely represent simply the effects of biological age, because males of a given chronological age are biologically about 8 years, older. So I'm not saying that there's no effects of sex, of gender. There certainly appear to be. I'm just saying there's a parsimonious explanation for much of the variation in terms of biological age differing between the sexes. And we can think about old age in relation to immune system effects on morbidity and mortality. So if we look at older people, without going into the gory details of the evidence that everyone, immunologists know a lot better than I do, older age involves reduced interferon production, more of a pro-inflammatory milieu, inflammaging, basic idea here. They also show increased coagulatory responses. These are one of the other main pathological effects that give rise to all the thrombosis and strokes and so on with COVID-19. And older age involves reduced adaptive immunity responses, which we see very clearly in responses to vaccination. So if you vaccinate somebody in their 30s or 40s, you're probably going to get a good response, especially if they're female. If you do it to somebody in their 80s, you're probably not going to get anywhere near the level of response. So we're going to develop these vaccines, and the people who would benefit the most from them are the people who they're going to work the least well in. Okay, but clearly more needs to be done in terms of effects of aging, the aging immune system in males and females in terms of understanding the pathology. Finally, a couple slides on virulence and the evolution of virulence. Evolutionary theory of virulence, we have a trade-off between transmission mode and rate and the levels of virulence in terms of mortality. So something like the common cold has low virulence because it relies on mobile hosts for direct transmission. If it had, if these had higher virulence and direct transmission, then the host would just get too sick to transmit very well. The host would stay home and not transmit. If you have a vector to move you, then the pathogen can build up to higher levels in the host, get better at transmitting, also get more virulent because the vector will move the pathogen for you. Now, SARS-CoV-2 is kind of making a hash of this, apparently, because it can transmit before symptoms appear pretty well. At least it appears to be pretty well compared to other viruses. And this is, once again, function of the effective anti-interferon system. In doing so, it can break the trade-off between high viral load and morbidity. So you can get a high viral load and be pre-symptomatic because your interferon system has not really kicked in properly and made you feel sick. Okay, So you can have a high viral load and good transmission directly between people. This is different than SARS-CoV-1 and MERS-2. These have much higher fatality rates, and they seem to make people much, much sicker. It's easier to identify the cases and isolate them. But this guy seems to be rather different in that regard. We end up with high transmission and low virulence in young people, and then of course high virulence in older people. And my last slide, we're getting back to Batural selection here. Here's a SIRS model. This is from some wonderful work by Troy Day and Saliato and others in the preprint server. What are we doing in terms of imposing selection on the virus? What's happening in principle with regard to natural selection, Batural selection, sorry, on the virus right now as a result of our public health interventions and any other sorts of effect. So here's a little SIRS model, susceptible, infected, and recovered, exposed. There's your pre-symptomatic period here, and you're getting symptomatic, and when you get symptomatic, then you get isolated, right? Okay, so what do we expect in terms of Batural selection? Now, we expect strong selection for a longer pre-symptomatic phase. And this is because as soon as somebody shows symptoms, they get isolated, in principle anyway. And when they get isolated, the virus is dead, in principle anyway. So you don't have selection operating anymore. Okay, you just have anybody with any symptoms, whether those symptoms would end up in high or low virulence, they just get isolated and the virus is dead. So we don't get selection against higher virulence here because with the public health system, the virus in tested individuals, they're fully isolated, they're dead anyway. So that trade-off between virulence and transmissibility is more or less broken, unless we have some sort of association between what's going on, the length of the pre-symptomatic period or the viral load in the pre-symptomatic period. If that affects virulence, okay, then that can be under selection and indirectly influence virulence, and that's how we could be getting selection on virulence right now. What about other possibilities? We can get higher virulence if we have a vector. Do we have human vectors? In Canada, we've had terrible trouble with people who have moved between long-term care homes and vectored the disease between more or less immobile people. And this could potentially select for higher virulence as well. We don't have direct evidence of that. And we may also expect selection for transmission of the virus from being present at lower levels in the environment because we're being so careful to wash everything and stay away from each other. We're selecting for the ones that are really, really good at surviving and getting between people. So that's all I have, and I'm happy to have any discussion, questions.
Okay, thanks, Bernie. That was great. Really nice. Meredith, do we have any hands that are raised?
No raised hands at the moment, but just as a reminder, you can raise your hand to ask a question verbally. Do that by clicking participants at the bottom of your screen and the raise hand in the panel that pops up. And it looks like we did have one person raise hand during that time. So we'll go to Mick Elliott to ask a question. Mick, you can go ahead with your question.
Hello, can you see me?
Yep.
Okay, I can hear me, okay. So we have these various factors which cause bats to be a dangerous harbor of human diseases. But this doesn't explain why COVID-19 is more dangerous than the other bat origin diseases that we've had in the past, like SARS and MERS. If these evolutionary processes have been going on promoting virulence in bat diseases compared to the human immune system. What makes COVID-19 so dangerous?
Well, the short answer is I don't know. The longer answer is that I would hypothesize that this particular virus is especially good at interfering with the interferon system, and as a result is better at pre-symptomatic transmission. And why it happened that way? Well, you really need to look at the natural host of this virus and see what's going on in terms of the interaction with the horseshoe bats that it naturally lives in. That would point you towards collecting those particular data.
Great, thank you. So from the chat box, Michelle Blythe asks, would you expect differences in virulence depending on the public health measures in different parts of the world?
Well, that would depend. And so at one extreme, you could have a very low level of public health in terms of isolation of infected individuals. And if that were the case, then you could still have the trade-off between virulence and transmissibility being operative, and you could select for viruses that were less virulent because being less virulent helped them to transmit better. Okay, so if you had quite poor public health, then you could, in principle, select for lower virulence. If you have very good public health, then as soon as somebody coughs or sneezes, you have them totally isolated, and you're not selecting for higher virulence here, but the point is that you're not selecting against it, okay, because those people are essentially out of the picture. And in those circumstances, the key becomes, is there any association between the events during pre-symptomatic viral load or the length of the pre-symptomatic period? Is there any association between what's going on there and virulence? So if you select for a longer pre-symptomatic period, that might end up selecting for a lower viral load, and it might evolve to be less virulent, or it could go the other way. You could end up with a higher pre-symptomatic viral load and end up with higher virulence. And so you can collect data on that by looking at what's going on in the pre-symptomatic period and how long it lasts and viral loads.
Great, thanks. So we have another question in the chat from Anna Clara Nadim, who asks whether the gender bias and mortality that you were discussing could reflect a difference in exposure rather than a difference in lethality itself.
Yeah, that's a good question. The data from British Columbia that I'm most familiar with shows that actually females tend to get it a little bit more than males do, and this seems to be a function of exposure and people who are working in healthcare. But there are plots that show mortality and also the overall age and sex distribution of people who have tested positive. And testing positive is fairly equal between the sexes, but the fatality rate is considerably higher in males, as we saw.
Great, thank you. And it looks like there's some discussion in the chat more about individual differences and sex differences. But we'll go next to Bob Perlman for a question. Bob, you can go ahead with your question. Bob, are you there?
Yes. Now am I unmuted?
Yes, you're unmuted now. Go ahead.
I'm interested in variations in transmission, and there's some talk about people being super spreaders. And is that because their behavior gets them to interact with more people, or do they have, is there something different about their disease that causes them to transmit to more people?
Well, I can only propose a hypothesis there, but you can think about people may differ in the efficacy of their initial response to the virus. So the initial interaction between the virus and the immune system may be such that with some people, their interferon system is relatively highly suppressed, so they don't turn on sickness behavior, but they end up with a high viral load, and those people could be asymptomatic and have a high viral load for a long period. Now that's quite conjectural, but the idea does follow, and it could certainly be followed up on. So the asymptomatic sort of super spreaders would kind of be one extreme of this type of effect of getting the pre-symptomatic transmission because of the interferon interactions of the bats and the viruses.
Thanks.
Great, thank you. So diving more a little bit into the asymptomatic transmission, Cynthia Beal noted in the chat that there was an article noting that COVID-19 is distinctive because it infects the throat and is transmitted before it gets into the lung and causes symptoms. So she's wondering, do we know why COVID-19 does this, and how does that figure into this model?
Well, the receptor is in a lot of places. It's not just in the lungs. It's in blood vessels and all over the body. One of the factors I haven't talked about at all yet, and it's another similarity between humans and bats that I think is relevant here. There's some evidence from rabies that bats can spread viruses via their echolocation systems, essentially blasting them out in their, the types of vocalizations that they have. And probably a lot or most of the COVID-2 transmission is consequence of the fact that humans talk and yell, as well as say coughing and sneezing. So this similarity may be important in terms of sort of the bat-human axis. And if we were looking at the evolution of transmission, say in the long term, we might think about, well, what is the role of transmission via the avenue of speech and loud speech. The data from choirs is probably the best example of this in terms of super spreaders, a number of cases now, one of them down in Washington State, where one person in the choir of dozens of people is sick, and it gets spread to most of the choir as a result of the choir practice. So vocalization is a commonality there, and that would probably be tied to the upper respiratory system, including the throat, and this could be another reason why this virus is especially bad. I mean, the reason these are bad, the SARS and MERS are bad compared to the other coronaviruses that make a common cold, is because they get into the lungs as opposed to just the upper respiratory system, and that's got to be some function of the receptors and the cells that get infected.
Great, thank you. It looks like we have a raised hand from Salih Khair. Salih, you can go ahead with your question.
Hello. First of all, I would like to thank you to this brilliant presentation. I'm not sure if you hear me properly.
I hear you.
We do.
Okay, thank you. I would like to ask that after people beat this virus, there could be some permanent damage even in young adults. And I heard some news, and there are some studies on it. Do we have enough proof on it, on the permanent damage after beat this virus?
Permanent, you mean permanent pathology in people?
Yeah, yeah. For example, last days I heard that after recovering, the coronavirus could anti-thyroid anticore in the human blood. There are some studies on it, but I'm not sure if it's I don't know if you get me.
Yeah, no, you would certainly expect that that would be that long-term pathology would be a consequence of the hyperinflammation and the hypercoagulation responses.
Or asymptomatic, for example, I beat coronavirus, even I don't know. And after 4 years, I have some scenarios on it, and we recognize that it's actually because of my coronavirus coronavirus history. If this scenario could be possible, I don't know if I express myself properly.
Well, no, I think I understand your question. I would certainly expect, I mean, that sort of effect just from the nature of the mismatch. I mean, we've got the ACE2 receptors in all sorts of places in the body, and we see all sorts of curious pathologies. So, I mean, that sort of long-term damage is not really going to be subject to selection. It's just going to be something that's a byproduct of this particular interaction that has all sorts of maladaptive elements for it, especially for humans. So I would say you would certainly predict it more with this virus than with human adapted viruses. Absolutely.
Okay, thank you so much.
Great, thank you. Next, we have a question from Melanie Martin. Melanie, you can go ahead with your question. Melanie? I know she mentioned she was having some difficulty with her mic. So I'll go ahead and read the question from her. Let me locate that in the chat. So Melanie says, the COVID-19 mortality curve does not match all-cause mortality if you include infants and children less than 5 years old. Are their interferon responses consistent with this, or do you think they likely are more protected from other mechanisms?
I would need to defer to an expert in terms of what's going on with young children, because they are in a very different sort of immune system related environment even than somewhat older children are. The infants, they're getting the protection through the milk, and they have an immature system. And the thing about young kids in general is that they are continually being exposed to these new diseases, so the immune system is expecting that. The main difference with the pandemic virus is that we don't have developmental novelty, we have evolutionary genomic novelty. So our genome doesn't have adaptations for this particular virus, and so infants would be more vulnerable in that regard. But the devil is in the details when you're dealing with this sort of novel and mismatched situations. So I can't really speak about that data or even what to expect. But the mortality distribution that we see overall is not that unusual for viruses or other infectious diseases.
Great, thanks. Next, we'll go to Kyle Summers for a question. Kyle, you can go ahead.
Hey, Bernie.
Hi, Kyle. How you doing?
I'm good. How are you doing?
I'm all right. You look like you're in Panama.
Venezuela.
Okay.
No, I'm fiddling with my backgrounds, but it's not working very well. Anyway, I had a question about, so like there've been a number of people who have argued that this can be viewed as a kind of cardiovascular disease more than a lung disease. And and that in particular, it seems to be releasing these, after binding to the ACE receptors, it's causing the release of these von Willebrand factors, which are proteins that are then causing these really nasty clots. I mean, you mentioned that the coagulation system is messed up, but I was just wondering, is there anything about the bat physiology that makes them less susceptible to this kind of coagulation cascade? Where, I mean, could that be another mismatch where?
Yeah, that's something that I did look specifically for, trying to get information about the use of the anticoagulant system in bats. And when you do any searching on that, all you get is vampire bats, because people have been so fascinated by the anticoagulants in those, and there seems to be absolutely nothing the use of that system in bats. So that really is a major research gap. And I mean, as far as the pathology goes, there seem to be these interactions between the high inflammation and the high levels of activation of the coagulation system. There's kind of a, positive feedback going on there, where the body essentially, self-destructs in certain tissues. But as far as bats, I mean, they're dealing with it, right? I mean, the bottom line is they can handle it, and we really don't know how they do that. So we should take some of this. I mean, I'm sure people are doing it, and it's very hard to do. But any more information we can get on how bats are suppressing the virus is crucially important. So if you're in Venezuela, you could go out and get some bats for us.
Yeah, I'm not, but not for a long time.
Bernie, if I could just ask a quick follow-up, sort of related to that. I think I heard you say at the beginning of your talk that bat viruses can tolerate a higher temperature. Is that and I don't know whether that means they have an optimal temperature for replication that's higher than viruses found in other mammals, or they just have a broader range of temperatures that they can withstand? I wonder what the evidence of that is, and if you or anyone else has any knowledge about that, because I was sort of surprised to hear that.
Well, that's based on the findings about what happens to the temperature of a bat when it's flying, and the temperature goes way up during flight. And conversely, they go into torpor sometimes when they're resting, and the temperature goes way down. So the idea about tolerance is just based on the logic that, well, if the viruses could not resist high temperatures during flight, they would essentially be wiped out as a result of the high temperatures. As far as direct tests of the temperature tolerance, in vivo of bat viruses versus others, I'm not aware of any direct information on that at all to speak of. And I don't know of any good evidence having to do with the role of fever responses in the pathology of the COVID-19. A lot of times with the older patients, you don't get much of a fever. Fever is expensive, and they just don't invest in it.
Okay, thanks, Bernie. I think we're going to have one last question. One last question at this time. Rosalind, I see your hand is up. Would you like to ask your question?
Can you hear me?
Yep.
Thank you for a very nice talk, Bernie. I just wondered if anyone's taken naive to SARS-CoV-2 bats and infected them to see what their phenotypes and behaviors are, because we don't yet know for sure that this is the originating animal, as far as I know.
Well, not that I'm aware of. I mean, I know there are a number of groups that specialize in that area, and it's certainly something that needs to be looked at closely. there is a diversity of evidence that fits with this virus coming from bats, and it would be very, very surprising indeed, I think, to find out that it didn't at this stage.
Oh, okay. Thank you.
Yeah.
Okay, since we're hitting the top of the hour, I just want to thank Bernie again for a fantastic presentation. I want to thank Meredith for keeping things running so smoothly, as always. And I want to thank all of you. There are a lot of questions still that haven't been answered, but I think this is a really productive start on this question, on these questions. And I really appreciate everyone's engagement. This is actually the 12th of these Club EvMeds that we've held, and we have 3 more already lined up. Next week, we'll be hearing from Barb Natterson-Horowitz. It will be a book club, Club EvMed, and she'll be talking about her book, Coming of Age on Planet Earth, The Nature of Adolescence in Humans and Other Animals. So please attend that. You'll also get a short excerpt from the book as part of that to prepare or just to have to read. After that, we have Verena Schunemann, who's going to be talking about paleogenetics. She'll be joining us from Zurich, from the Center for the Institute for Evolutionary Medicine in Zurich. And then we have, after that, Nina Jablonski will be speaking about the evolution of skin color. We don't know exactly what week that is. It'll be July 13th or July 20th. And we also have Lou Muglia and some of his colleagues who are going to talk in late July or early August. So stay tuned, join our email, well, I guess you will be on our email list now. You can easily opt out, but stay tuned for more of these, and thank you all very much for attending. This is really great. Great to see everyone. Thanks.
I wanted to thank everybody, and Charlie and Meredith, and I'm happy to talk on email or other ways with anybody who wants to chat more.